thank you all for joining. I would like to take, let's say, 20 to 25 minutes to give you a roundup where I think AI currently is making the biggest impact in life sciences. And yes, I'm doing this from Half Moon Bay, as revealed by my background. So I am in California for the moment, but usually I am teaching in Belgium at Ghent University. So just one slide on my background. I don't want to dwell too much on it, but I'm a chemical engineer, did a PhD in signal transduction, apoptosis, cancer research. And then on the right-hand side, you see my academic footprint, which was largely based in Europe, taught in Amsterdam, in Sweden, but mostly now in the University of Ghent. And on the left-hand side, you see that I've been working in biotech companies. The common theme there is diagnostics and genetics, epigenetics. So we moved now everything that I do in entrepreneurial-wise into an investment fund that is focusing on big data and applications of AI in life sciences. So I think in that respect, very appropriate. Just to give you a flavor, and that's really the last introduction slide, these are the companies we just invested in. So instead of having to start companies in a serial fashion in a fund, we can do it in a parallel fashion. You might recognize DocAI. I'm not going to cover a lot of what's being done there, but people with a healthy interest in AI and life sciences definitely should check out what DocAI is doing. They have a very interesting offering there. All right, let's dive into it. So I want to start, since I'm in California, with Maverick. It's an example that I took from the book of Topo. A little kid with very asymptomatic seizures, not something that you would immediately be able to diagnose. And that's what happened in this case as well. The seizures started happening after a week. And it wasn't exactly clear what the problem was, and classical diagnostics was out of options relatively fast. So in this case, they did the whole genome sequence. And what I want to show here, because I get often asked, like, what's the point of, and why is the future of your health, as Nebula Genomics is putting it now, in sequencing the full book? Is it all clinically relevant? And I think this example shows that they found an enzyme in a biosynthesis pathway that makes an essential amino acid. So there was a mutation there, which was found by whole genome sequencing, which lacked the ability, or gave, the kid couldn't make the essential amino acid to come up with essential proteins, in this case, especially neuronal development. Once that was diagnosed, it was super easy to treat. You just supplemented in the diet. If you supplemented in the diet, then the kid was totally normal. So you can say, well, this is probably something that is exceptionally rare. And that's where I want to come with this slide is, if we would sequence ourselves, 3 to 5% of the people would have to go on a drug. They call this incidentalomics. So if you do incidental findings, which would not, you didn't have a clinical reason to start doing this, then you would come up with, again, 3 to 5% of the population that would benefit from starting a treatment immediately. I want to make this point a little bit more elaborate, and then I'm going to shift gears. So you see here kind of the biology revolution, and why it's a revolution is that we can, of course, do the DNA on genome scale, which is the top left corner. But the top bottom corner, we can also do RNA, and we can actually do protein omics-wise. And I don't mean mass spec and very complicated. You can turn all of these different facets of biology into a sequencing high throughput effort. The middle part, which is the regulation, it's kind of the conductor of the orchestra, also can be done by high throughput methods. And the metabolome is finally also coming to age. So we can measure small chemicals also on a comprehensive level. So what I'm saying is everything that we figured out the past 20 years in terms of next gen and third generation sequencing is also revealing all the other molecules that matter in life sciences. So including RNA, proteins, and the metabolites. That's all there is. Metabolites, I mean sugars and lipids and cholesterol, for instance. And so all these things can be measured at scale. All right, so let's shift gears. I'm sure you're familiar with bioinformatics being the center of the universe. which is what I teach. But I want to coin something here, which is discovery informatics. And that's where I think AI is going to come in interestingly. I'm sure you're familiar with protein structures, and there is competitions where people try to predict protein structures. So they have a crystal structure, keep it for themselves, and then they put out a competition. And that competition was won last year by AI, by Google. DeepMind was actually acing all the other contenders and came up with the most accurate protein prediction. So imagine this, what we typically do in drug discovery or in pharma biotech is we do this. We say this protein is involved in a given disease. That's our hypothesis. That we collect biotech data or screening data, and then we're going to analyze the results and come up with a new thing. So what we can do actually is we can automate this total cycle. So what you see here is Eve, which is the second generation robo-scientist. The first one was called Adam, but Eve was better. And what it basically does is it's going to execute experiments automatically. And what's happening is you have your protein structure that I showed is also coming from an AI. This robot is going to execute an experiment to see how to modify the activity of the protein. That's generating data that is analyzed by AI and is going to generate hypotheses to do more focused second iteration drug screening trials. So what you see in drug screening is that humans are totally out of the loop because they become rate limiting. And what you see happening is that knowledge is acquired by analyzing data, proposing experiments, the robo-scientists executing the experiments, generating new data, and you get into this loop where actually humans are out. And this recursive iterative screening is something that pharma and to some extent biotech is quickly adopting. We made versions of this. This is just a little toy program where we did we programmed an ability to spawn in Minecraft any type of protein. So this was a lipoprotein A1, but we're still toying now with it. You can spawn a protein, but you could also start gamifying drug discovery in Minecraft. It's something I won't go into, but inhibiting a protein is trying to throw things at it that stick, basically. So that's something we can easily turn into a little computer game. And since I'm from Belgium, I wanted to share one poem from Belgium. And I actually found an English translation, which is between the dream and act, there is always hindrances and laws. So between this cycle of coming up with new drugs and applying AI to predict proteins and structures and functions, one of the biggest hindrances is actually clinical trials. So before, as we know, before we can apply some of our knowledge into real clinical settings, we need to show proof that these things actually work. And I think clinical trials is the area where AI is currently making the biggest impact. Clinical trials, typically, this is the cornerstone of discovery. It's coined in the 70s, called randomized clinical trials. Control group, and then a group that gets an experimental treatment, and you try to compare results there. So what is so bad about this, and what can be improved? So what can be improved if you have two companies or two drugs? You see that you're stuck with two randomized clinical trials, each having a control arm. It does make a lot of sense to start sharing the control arm, right? So at least 2/3 of the people are getting an experimental treatment that is now being compared to the standard of care group, which is typically the control arm. But imagine this, that I can turn this into what's called a platform trial. And I only know of one disease where currently the platform trial is being adopted, and that's glioblastoma. It's a brain cancer that is relatively rare. So what you see here is that now we get therapies within one trial being combined and using a shared control arm. Of course, we can turn this into what's called an adoptive platform trial. We can now phase in an ongoing trial new arms and recruit patients specifically to a new experimental treatment, which means that we have to find ways of also test for non-futility and closing arms. So what I think is going to happen here, and it's going to be adopted relatively fast the coming decade, is we will have within one disease area actually one continuously ongoing trial that is not going to be closed. It's going to be reported on certain endpoints, but it's going to keep going dynamically with including new arms, finding diagnostics to recruit patients to those new arms, and come up with very sophisticated statistics and AI to randomize patients across this moving beast. Shared control arms, and then I'm going to go into what probably a lot of you heard about, digital twinning and synthetic control arms, is something we can also start doing using AI, and I will come back to that a little bit later. So what needs to happen to make this a reality is you have to come up with Chinese walls, because here you have competing therapies, competing companies actually sharing resources in one common trial. So that's something with the software world has figured out a long time ago. We have all these interesting licensing models where people can protect still what they own and still be cooperative or not cooperative, or infectious or non-infectious licenses. I'm sure pharma has to start to be opening up a little bit on how they license and protect their assets. There's been reviews published on what pharma is going to evolve into when all this clinical trial is coming to age. I want to say and focus on one thing, which is the bottom sentence. There is a pretty open communication you can have proactively with FDA on how you want to conduct your trials these days. And the keyword there, sure you heard about it, is real world evidence and real world data. And I want to give you an example, 2 examples on what it means and what it could mean also now in Corona times. If this would already be implemented, we would be able to advance much, much faster. So this is an example. I'm sure you heard about Enbrel. It's a drug from Pfizer that was found in insurance claims to postpone or maybe have an effect on the development of Alzheimer. So this is a rheumatoid arthritis drug. If you just look at insurance claim, you see that people on the drug develop Alzheimer much later. So what you need, in this case, it was actually confirmed by the science, because the underlying pathways that are resulting or thought to result, the inflammation pathways in RA, rheumatoid arthritis, are also implicated in the early development of Alzheimer's. What you need is not this sophisticated AI. And that's an important point. We have all these very sophisticated methods on supervised, unsupervised, all different kinds of methods. We just need a very simple 2x2 table to see, is a certain drug maybe associated with a certain clinical phenotype. And all that relevant clinical data is the problem. It resides with different companies, insurance companies, but also sits in different EHRs or MRRs, so medical health records, MHRs, EHRs. So we need relatively simple statistics, but we need to get our hands on that data to really make that happen. So I wrote a paper on how maybe blockchain technology can work in that, but I want to apply this to Corona. So imagine this. So this is what we know about the coronavirus infection. So coronavirus is recognizing a very specific receptor, the ACE2 receptor on the pneumocytes. And when it's recognizing the receptor and enters the cell, it needs to be cleaved. And that cleavage is happening by an external extracellular protease called TMPRSS2. So one of the cool things is, if you have the receptor, you recognize the coronavirus. But if you don't have the TMPRSS, you don't have the protease that's going to cleave, you don't get entry of the virus. So here's the cool thing. The cool thing is there are drug therapies that are actually regulating that protease. There are therapies called androgen deprivation therapy that downregulate the TMPRSS. And that's typically given to, for instance, prostate cancer patients in a very late stage. So what people started noticing just by accident, and that's the frustrating part, is that actually advanced cancer patients were doing better than expected when they got infected with corona. So that's a very simple 2x2 table. And the underlying biology is very well understood, because they get androgen deprivation, which downregulates the TMPRSS, which doesn't allow the virus to enter. So that's a very, very simple thing we could have learned systematically by looking at very simple outcome data. A validation trial is, okay, so prove your point by showing that people that are on androgen deprivation therapy are doing better on COVID. You could say, well, there are actually people going for, let's say, chemical castration. The transgender community, for instance, is often exposed to that. Are they doing better? That's a very simple query to write, and that could validate independently this hypothesis. It's something that is now just not available. It's not something we can do. So on the therapeutic side, we could learn and advance much, much faster if we would have ways to do these very simple 2x2 tables by unlocking that associated data. So this is an example on the diagnostic part for corona. I'm sure you're familiar with the RT test and sending your samples over, drive-throughs, all that. So what we're getting to in diagnostics is we need also ways to faster personal testing and to solve two outstanding issues there, which I think also AI will help in. That's remote sampling. Part of the reasons we need doctors or nurses to be present is to make sure we are not probing our ear to get a negative test result, and we are really probing the sites where the virus could reside. So that's called remote sampling. AI could definitely help to build in controls that we are sampling correctly. And then we want to be able to leverage edges being your devices, but also very simple methods that can allow me to generate a result where I am within a time frame of, let's say, 5 minutes. I don't want to do PCR because then I need machines and specific hardware. I want to use methods like graphene, which allows biosensing without amplification, just at a constant temperature. So there are methods that are coming along and coming to age that would need to solve that remote sampling and that detection part. That's also happening more or less in real life as we speak. So I want to get to this personalized medicine by coming, let me see the time. Yeah, I can do that. So you know this, right? Personalized medicine is the right dose, blah, 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 for the right patients. So this is an example that frustrates me enormously. So this is a P450. So when you take a drug, your drug is metabolized in your body, and you need enzymes to do that. This is the protein enzyme called the P450. We know this very well. There is actually two, like CYP2C9, and there is the VCORC1. And they, if you have very specific variants, play a role in how you respond to, in this case, warfarin, which is a blood thinner. It's what we use to poison rats. That's what we do. We bleed them basically to death. If you have, as a person, certain mutations in these genes, you're going to require a different dose of warfarin to be healthy if you want to thin your blood, given specific indications. That biology and that pharmacogenomics, as we call it, is extremely well understood. It's so well understood that it's actually mentioned in the insert of the drug. Still, when I'm going to the drug store, nobody's checking whether my genome, which I might have from 23andMe or whatever, is compatible with the drug I'm taking. And the reason is that nobody's really motivated to do a better job there. The pharmacist is box moving. The doctor is saying, if it doesn't work, you're going to come back. You're really going to waste my time in explaining you basic pharmacogenomics biology. So one way of approaching this is putting the patient in the center and empowering the patient with apps and tools that allow him to take control over whatever he's doing. So that's this patient centricity, and I think DocAI is a beautiful example of what can be done with current technology. So we know, and I'll give a few more examples, I have 5 minutes more to go, that AI can definitely outperform doctors. But it's this black box that apparently patients are not so eager to trust. So what can we do about it? So this is an example of, famous example of an ophthalmologist trying to look at an eye and trying to say, is this a male or a female? So this example actually shows that the most advanced human expert cannot see based on the eye iris picture, whether it's coming from a man or a woman. AI can, so whatever, this is just a Google example, can very accurately do that. And now we get into that area where you say, are we happy to be doing this with a black box, or do we need to understand what the AI is doing? So of course, there's lots of methodologies calling explainability of AI and trying to say, what can we learn from how the AI is doing it? And this, in this case, it's not easy. AI is just able to look at multiple dimensions at the same time. And it's coming up with that very accurate prediction by combining a lot of features that the human mind is just not capable of doing. Of course, the AI is just a toy example. Sorry, the eye is just a toy example. It becomes more interesting when we look at, let's say, radiology images and multi-parametric MRI imaging. There is no cancer where AI is not outcompeting today human radiologists. So this is just examples from the last year where in breast, in lung, and in prostate, AI is outperforming because it's just able to look in all these dimensions at the same time and learn from many more images and cases than what a human residency would ever allow you to do. So you probably are aware with this Algorithmic Accountability Act, which is kind of leveraging some of the concept of GDPR in Europe. And actually, AI is being approved by FDA. Currently, there is 60 algorithms that use AI-based methods that are approved by the FDA. And I think what we need is, again, that same insert I showed you before. We need an insert for an AI algorithm that is basically stating all the things we expect from a normal therapy, meaning all the control data, all the reproducibility, especially when it's adaptive AI, making sure we keep tests to keep the AI in check, that it's not converging to something that we didn't intend it to be. So all of these things that we do for regular treatments, we need to find ways of mimicking that in the AI world. But I want to leave you now with two thoughts that I think might spark the discussion afterwards. One is, you all know GPT-3. GPT-3 might be allowing to pass the Turing test. I think in the paper they get like 85%. But what I'm interested in is AI with a totally new defined Turing test. So you probably read about an AI being able to rediscover that the Earth orbits the sun, right? Just from looking and leveraging a lot of data. You need an AI that is doing more than building language models. You need an AI that is going to also be able to infer logic and build methods and relationships between terms and ontologies and all. What would it be? What would it take for an AI to rediscover basically current molecular biology? Can we give, or that would be for me the ultimate scientific Turing test. Can we give the book of life being the genome, and would we have an AI that rediscovers the fact that, hey, there is coding parts, there is proteins. And I think what we definitely would see with an AI that is able to do that, it's going to rediscover, or not rediscover, it's going to discover a lot of new things, because there's a lot more than the simple central dogma happening with genomic data that we don't know about. And then the last thought really is that everything in technology and AI is moving towards single cell and single molecule. And one of the things I'm interested in the lab is I want to build new organisms that are actually combining not just molecules in synthetic biology, but I want to create new organisms by combining cells that we reprogram or make synthetically or harvest and reprogram from different organisms into a new, what's called xenobot. So a xenobot, or a living robot, is not composed of silico components, but it's built of living components. So we are currently working with microfluidic and AI methods to come up with predicting on the left-hand side what the behavior, emerging behavior of such a new organism, if you can term it like that, would be, and then build it in real life. And what we build in real life, we measure with all kinds of functional assays and feed our simulation and AI programs with. So this is, I think, the most advanced things we try to do. We call it for the moment Xenobot. There's only 1 or 2 papers about it. And it's like reconfiguring life as we know it. But this is, again, we can debate for hours on what we can do with this and what this could mean for science. So that's what I want to leave you with. So I'm all the time in the world to answer any question you might have.
I see questions from Neeraj. Neeraj, can you ask your question, maybe to just unmute yourself?
Yeah, sure. Actually, maybe the most relevant question is this thing about xenobots. Is the work around discovering xenobots very similar to the work around, like, predicting proteins, or but just at a larger scale, or is it like very different kind of techniques that you're using?
It's very different. So what we do there is we need to first of all define the building blocks, which are cells, different cell types, and what their behavior is. And what they did in this initial paper is they combined sensor type of cells with actuators type of cells. So you want this thing to move in a certain direction and to then do certain things. So what you essentially do is you run big simulation engines and genetic optimization algorithms to come up with what configuration of what type of components could result in a behavior you're interested in. So it's more, it's very different than, let's say, the protein modeling part. The protein modeling part is leveraging more, let's say, classical sequence text-oriented bioinformatics approaches. There is no text going into the simulation of these xenobots. Proteins are essentially texts, right?
How do you define like stable configurations of cells? It doesn't seem obvious to me. I mean, I don't know much anyway, but how do you like discover stable configurations of cells that to attain tasks?
Yeah, certain cell types are able to adhere to each other. You can, of course, so they stick together. They have like, extracellular matrix, common receptors, and so they recognize each other, and that's how they actually come up with a multicellular state. So you can engineer them to stick to each other, or from organisms where they coexist that they recognize each other. There's also an auto-assembly part in it. So cells can stick together and they form layers like biofilms, which is something you can easily or relatively easily model. And then you can try to combine different cells. There are actually organisms. There is one very interesting one that is Dictyostelium, which is usually single cell. And then depending on an environmental clue, these cells decide to come together and form a multicellular fruiting body. And we know how this fruiting body is formed because it's following this external clue, which is cyclic AMP. And actually one of the coolest things is we can play with the external clue. We can seed your medium with very specific dose gradients of cyclic AMP. And then we can shape the form of the fruiting body. So we understand the process on how the cells are coming together in order now to interfere with it and to turn that fruiting body, which is typically ball-shaped, into a different shaped form. So in some cases, we understand that so I think it's not to be underestimated, this cooperation between cells and how we can manipulate that. And maybe to round up that thought, that's essential also in cancer, right? A cancer that is forming a multicellular clump is harmless. It's benign because it stays in place. But the moment the cancer can move into wandering cells that go to a different place in the body and leave the bloodstream and start rebuilding themselves somewhere else, we get metastasis. We get into issues. So that whole field between unicellular and multicellular processes and try to understand and manipulate that is really key in a lot of essential processes. I can perfectly imagine, for instance, having cells that recognize fat and combine it with cells that dissolve it and have that in your bloodstream, clean up your plaques and avoid atherosclerosis. An obvious application is cells that would aggregate microplastics and be able to handle microplastics, for instance. There's phytoremediation applications. There is lots of things we can think about where you want to combine functions that are now residing in different organisms. So that's something that is coming.
I do have a question.
Yes.
I've been trying to ask this ever since you asked for them, but it's been hard to get a word in edgewise between you two. So one of the easiest or perhaps most nearby things that we could begin to build with synthetic biology wouldn't necessarily be something that's never existed before, but something that has that we can't necessarily create. So a good example might be replacement tissue if someone's lost some tissue, or even a collection of tissues or an organ. So for example, if someone's been burned badly and they need skin grafts, or if they've got heart failure, you could replace their heart. That's really what I'm the most interested in. And I'm wondering, where in the synthetic biology field would I find the people doing that?
Yeah. So this is a field which is, thank you for that question, is called tissue engineering, tissue re-engineering. And I think you're absolutely right. For instance, the burned skin, we typically took skin from somewhere else and transplanted that. But what we can do today, and I think there's two interesting things I can say there, is called, when you take, you don't have enough skin cells to transplant. So you need to take a cell from the patient, like a blood cell, and then turn that into a skin cell. So it's called differentiation. So what we used to do is we take blood cells and we induce pluripotency. So it becomes more omnipotent, and then we gonna differentiate it to become a skin cell. And then we grow the skin cell ex vivo enough to transplant back to the patient. What is cool today is that current knowledge allows us to do something called transdifferentiation. So we can take the blood cell and reprogram the blood cell by doing epigenetics to become a skin cell, and then grow the skin cell ex vivo and transplant it back. But here's an interesting idea to think about, is this whole technology about ex vivo growing cells and transdifferentiation, which is cutting edge molecular biology and synthetic biology, reprogramming genomes, is actually done now in food industry. So if you look at people growing artificial meat, that's exactly what they do. They reprogram cells to become muscles and fat, and they reprogram them ex vivo. And that field is taking off in the Beyond Burger way of things. And it has to do with the regulation that is more cooperative in that area than it is in the medicinal field, because you are ex vivo growing cells. And again, cells don't grow, they die. And if they grow, you call it cancer. So there's a lot of safety things in, let's say, the medical part of it that we need to keep in check. But it's definitely, I agree, if people lose, for instance, their ability to make insulin in diabetic patients, let's just take a cell, a beta cell for that matter, from that patient, and expand it ex vivo in a petri dish, or reprogram it that it's producing more insulin, and then put it back in the patient. So those are definitely things people are looking into and working on. So taking the cells, the patient's own cell, their own genome, because think about this, we have 3 trillion cells with all the same genome, but they do very, very different things because they have a different level of regulation called the epigenetic layer. And we can now do called epigenetic editing that reprograms these cells. So we can take a pancreatic cell from a diabetic patient, turn that into a beta cell that is normally producing the insulin, and then put the patient's own cells back. It's just that we need to go through a lot of regulatory hoops, which the people that are growing the burgers in plates don't have to go through. So, but that's definitely something that is, I agree, an entire field of tissue engineering that is exploding.
So if the most common cause of death is heart and lung disease, are we seeing moonshot efforts to create artificial hearts and lungs?
Yes. So artificial hearts is something that are definitely a lot of groups are working on. I think most, again, 40% heart disease, 40% is cancer. So I'm not sure about the lung, but I think what the example I gave with the xenobot for atherosclerosis is keeping the arteries basically open. Definitely would also have an impact on congestive heart failure and heart attacks. But keeping the plumbing, which I call the plumbing, is the cardiovascular part of your body open and free and working, is definitely something people are working on. There are very good efforts in making hearts pump in a petri dish. So they start to combine different cell types that make up an organ like a heart and make an artificial organ that has heart-like behavior. I don't think the next 5 years or so it's going to be good enough to replace a human heart. We're probably better off with with heart transplantation and making sure we can get the transplantation issues solved. But keeping the arteries open and clean and the piping working is something that I think is going to be doable in the near future.
So if I were a young person looking to make my way in synthetic biology as an MLE, would working on these projects be a good place to start?
Yes, I think definitely there is a lot of applications and a lot of basic knowledge that will be generated by applying, let's say, AI, big data approaches. Yes, definitely.
I would love to give you my contacts so we could stay in touch and I could hear some more about like where I should look for work and stuff.
Yeah, no problem. It will be my pleasure.
You can also join our Slack community. Wim is there, so you can.
Yes, I joined it. Yeah. No, more than happy to pass on references. This is an area where I think we are in need for people that have, let's say, an interest in current computer and methods and applying them, data sets that are used in these applications. Definitely.
Do we have other questions?
There was a question in the chat from Stephen. He asks if there are any commercial companies that are using AI, I think for the same kind of use in medical imaging, or whether it's still in the research phase.
Oh, there's tons of companies. There's tons of companies that are doing, definitely for interpretation of images, like all the major suppliers. There's like Philips and Siemens and General Electric, and yeah, they all have what's called digital pathology and software that includes AI components. The keyword, if you want to be, let's say, at the cutting edge, is called multimodality testing. So where you combine these AI modules that run on images with everything else you know about the patient. And everything else means clinical, clinical features about the patient and the disease, but also molecular. So I think the cutting edge is combine molecular data with imaging data with clinical data. It's called multimodality testing, and you have to come up with models that can handle that. But no, there's a ton of companies working on AI and also edge AI. I know people, for instance, that are making devices to check if egg cells that you want to harvest for IVF are mature. So they actually are probing at home, real live, whether the egg cells are maturing, and they need AI, not transmitting images, but also on the device itself, to show that you took the right image, that you're looking at the right type of data to infer fertilization potential from. But you know all these examples of people taking pictures of moles for skin cancers, and so all these things are currently being pursued for sure.
So, question from Eric: what are the dangers involved with this type of research? Is there pushback like with GMO foods?
Yeah, the dangers are eminent, right? It's yeah, you don't need to be a master brain to come up with, if we can do this synthetic biology and genome editing and DNA assembly, and we have DNA printers, and some people might get inspired by genomes that are available. If we look at the corona genome, it's not super hard to make that and tweak that or do something with it or come up with variants of that. So yeah, definitely there is a lot of potential for using this in the wrong way and actually not. So the thing that I'm not sure you're familiar with Nick Bostrom's, it's called the vulnerable world hypothesis, where he says we make inventions, and we invented, for instance, nuclear power, and apparently, so it's not easy to get the power out of an atom, right? It takes significant equipment and fission or fusion reactors, but to get a genome to drift into a direction you don't want, and we have an example now of a genome that was been able to paralyze in the world in a question of months. So that is not too complicated. It's not too complicated to do in your garage a little genome editing. It's something that doesn't require a lot of money and not a lot of equipment, and it's very hard to keep in check or to control or to have to or to work with. So that vulnerable world hypothesis, where he says most of the inventions are white balls, they're fine. Some of them are black balls, which is nuclear energy, but maybe we're going to have inventions like genome editing, which are so commonplace that they can be, they can be hard to control. So there is definitely a lot of ethical issues that we have to think hard on how we keep that contained.
But there's no one solution, right? At some point, it will be just who has more power and advantage, basically.
There is.
Opinion.
Yeah. But so here's another thing, another way to think about it. You probably heard about this treatment for spinal molecular atrophy, which is, I think, one shot when you're a young baby. So if you get SMA, you die before the age of 10 because your muscles aren't growing. So the treatment for that is a CRISPR-Cas, an editing. So you basically repair the wrong gene. So one treatment is costing you close to $2 million.
Mm-hmm.
While I can order the CRISPR, so I can order the CRISPR-Cas online for, let's say, $100. I can make the guide RNA, so I sequence my gene. The guide RNA is also a few dollars. So what is, so the potential of there is the potential of a cure that is either going to cost you 2 million, which you might have or not have, or it's not covered by your insurance. I'm not sure how these insurance policies will evolve, but those numbers are quite astronomical. And there is the potential to just say, well, you're either going to die or you have to play in your garage with something that might work. So there are people, there is this whole biohacking community that is starting up, that is trying to do these things uncontrolled, which I'm definitely not advising to do. But how are you going to contain something that is either totally out of control in terms of price versus something that people want to democratize? And like we have open source software, we have this open source biology movement and do-it-yourself biology movement that allows basically people to edit their genomes. And people are saying we need that capability. There is a very cool Netflix series called Unnatural Selection, which I think outlines the current state. And I think it's a very well-made overview of the ethical and moral dilemmas we're facing by keeping this all open. I'm a bioinformatician. I not want to reinvent wheels. I want to reuse code. I want open source. I like all of that. But think about, with the current capabilities in biology, we have the same movement that is there. How are we going to keep that all contained?
Wim, with COVID, this kind of vaccination topic pick up, and everyone is like kind of waiting for this vaccine as, something that can help the entire humanity. What's your take on how AI is kind of contribution of AI on developing new vaccines and maybe deploying them much faster?
Yeah, we have a company called MyNeo. You can definitely look that up, myneo.com, where we also use AI methods to predict when a virus is there, like the coronavirus. What are the most optimal epitopes? What part of the genome is perfect to get your immune system activated, which is basically defining the success of a vaccine. And we participated in many of these vaccine efforts for cancer, but also for the coronavirus. So AI is the reason why we were able to come up with candidates from, the genome was published the 12th of January, and we got the candidates, let's say, within 3 to 4 weeks, and that's because of AI. And then we had to produce them. And again, now that RNA production has its benefits and downsides, it's easy to scale, but then you have this cold chain problem, and you can make probiotic versions of it, you can make protein versions of the vaccine, and they're all going to come. So I'm very hopeful, given the initial efficacy rates that we've seen, that this is a virus that is going to be susceptible to vaccination countermeasures, which is not always the case. We haven't solved that for HIV, for instance, which is also a virus, which is not something we can control with a vaccine. But every vaccine that is coming definitely had AI being part of its discovery. That I'm 100% certain. We come up with these epitopes by using AI methods. And for instance, to make it very tangible, what is the AI we're using is, when we have a virus, the body is trained to recognize non-self foreign bodies. That's basically what the immune system is designed to do. So how does it do that is by exposing part of this genome on very specific molecule called MHC molecules that are putting out a very specific part of that coronavirus genome that is then offered to the immune system to be recognized and sniffed out. So that potential of an epitope, which is part of the foreign genome, to be exposed on that molecule and being sniffed out by your immune system, it's what we call immunoexposure and antigenicity, is something we can model with AI. So we are able to leverage data. We have mass spec approaches to see what is being exposed, what results in an immune system, and we extrapolate that with AI methods and adversarial convolutional deep neural nets that we feed data with, and then we try to say this one is clearly the one that is going to be giving you the best chance of an immune response. And all of the candidates have gone through that. So the good news is there's going to be vaccines. I'm not sure how long the immune response is going to be there. If we have a new case of influenza where the virus is mutating and we need to come up on a seasonal effort doing this, that's hard to predict, but we'll keep monitoring the drift of the genome and see if it's going to escape the initial or modify the initial epitopes. Yeah.
Wim, I'm also curious. So coming back to that slide when patient is in the center. Like, let's say if I have like report from 23andMe or like another similar company, and I come with this report to my doctor, like kind of requesting I want kind of more personalized approach, to diagnosis and like drug prescription. Do you think it's possible today? Is there like people practicing these kind of things?
Yeah, that's a good question. I tried it multiple times with my doctor, but he's like, "What the hell?" It's something that, the system is not very receptive to. What I usually get is like, "Yeah, can we trust that data?" But yeah, of course you can trust that data. That data is generated with the same machines and the same methods as if I was going in for a genetic test in a hospital. So that data is totally trustworthy. Is that usually they don't know how to react to it. So they are very defensive, typically doctors. I had to take a statin, which many people have to do. And then there are certain SNPs in your genome, like a 23 genome, that predict whether I'm going to have side effects. So I wanted my doctor to be aware of my genome to prescribe me the right statin that minimizes side effects. And the doctor was basically saying, you can say whatever you want. I'm going to give you the cheapest one that's reimbursed the most. And if you have muscle pain, in 3 weeks, you come back and I'm give you something else. I'm not going to base my decision on your whatever file. And he has a point. It's like, how do I even know that what the data you're showing is yours and not from your sister? Which, again, if it's my muscle pain, I have a direct interest to use the correct data. But there is, of course, ways of misusing the system to get, for instance, higher dose of a certain medication. I might say I'm a poor metabolizer. Look at my genome. I need higher doses, just to get higher doses prescribed. I could imagine in the future people misusing the system that way. So we need a way to get chain of custody but I think technically, if that's doable for a bank account with money, it should be possible to be with medical data. I should be able to say it's my sample taken by that person. That whole blockchain thing where I keep track of, it's done by a medical testing center that is following procedures to minimize sample mix-ups, right? I can very easily imagine a world where I'm a doctor. I can check whether that data that's being shown to me is probably indeed the data that I'm required to make a decision on. but that's not something that is there yet, and that's frustrating. So I think the biggest eye opener is that adverse drug reactions are the fourth biggest killer in the US.
Mm-hmm.
So if you're able to minimize that even by a small percentage, I think someone in that value chain should be interested.
Yeah.
Even if it's like the insurer, like, yeah, they should be able to get better at it. Yeah.
Do we have more questions? I don't see in the chat. Yeah, I think we're ready to wrap up. If you guys want to connect with Wim, asking questions, feel free to join our Slack community. I'm again posting this link. Yeah, thank you, Wim. It's such a, like there's so many things to discuss. It's like,
yeah,
feels like a little bit overwhelming, but thank you very much for your time.
No problem. It was my pleasure. And yes, and maybe one last reference, if you look for that patient centricity, I think doc.ai is not a bad place to start. They also embrace that method, and I think they are very advanced in that. All right.
Yeah, thanks a lot. Thanks a lot, Wim.
My pleasure. Have a good one.
Yeah, guys, feel free to join our Slack community.